Hello my friends, welcome back to my channel. Today I will continue my previous work about genome-wide gene family identification. After we retrieve the gene sequences of the gene family, now we will study the protein properties. So please, for who is interested in the study genome-wide gene family identification, follow me to study the protein properties, how we can make prediction of subcellular localization, how we confirm the sequence we obtained. In last lecture, they are open reading frame, and also we will study the prediction of molecular weight and isoelectronic points. Please join me in this video and let's get started. In the genome-wide gene family identification, the first result normally is a table that includes the molecular properties of the gene family members. Normally, this table consists of the generic name, so you can mention the name based on other references, or maybe you can give the name by yourself. Also, you can, as showing here, you can see the subcellular localization. Is it like a prediction? We will study how we can use different bioinformatic tools to predict the subcellular localization of these proteins. Also, maybe you can find the accession number in different database, and also you can find the operating frame length. And today I will explain what is the operating frame and how we can predict the length of the protein or the gene sequence. And also you can find some information about peptide lengths, the number of exons and introns, the molecular weight, and isoelectronic points. This table actually is very simple, and we will show you step by step how we can construct this table. So my friend, as I told you before, the basis of the genome-wide gene family identification is using protein conserved domain to retrieve the protein sequences. So the first step after retrieving the protein sequences of the gene family members, we have to confirm that by using some bioinformatic tools, really they have the same protein conserved domain. So I will show you later. And next step after retrieving the cDNA sequences and confirming really the protein sequence have the same conserved domain, also we have to confirm the CDS sequence have ORF. So what is ORF? ORF is open reading frame. This open reading frame refers to the portion of a DNA or RNA sequences that can be translated into a protein. So it started from ATG that could to methionine residue and ended by one of the stop codon TAA, TGA, or TAG. One of the most important information can be added in the table is the prediction of the subcellular localization. What is the subcellular localization? Study the subcellular localization is of significant importance in understanding the function and the regulatory mechanisms of our protein. The genomic DNA is in the nucleus, and after the transcription, the messenger RNA can be translated and can be translated and in the outside of the nucleus in the cytoplasm and in ribosome. After protein synthesized in ribosome can be derived to different compartments like mitochondria, Golgi, chloroplast, and vacuole. And these proteins, even they have the same conserved domain, but maybe have different function based on the compartment, and maybe can involved in different cellular meta processes. So the studies of subcellular localization is very important for interpretation of our result. So to understand the subcellular localization, we know that the protein is synthesized in ribosomes, but how the protein are targeted to different compartments. Actually, after protein synthesis, The precursor protein consists of transient peptide and mature protein. So the transient peptide, also known as targeting peptide or signal peptide, is a sort of sequence that exists in the N-terminal of the protein. So the transient peptide play a crucial role in protein targeting and the localization to specific subcellular localization. As shown here, after the protein synthesized in ribosome, so the precursor protein consists of the transient peptide and messenger protein. So this transient peptide will, by some complex mechanism, can target the protein inside the different compartments. This example for chloroplast. So after driving this protein to the chloroplast, so the transient peptide will be cleaved and just a measure protein will be exist in the compartment. So by using some bioinformatic tools, we can predict the transient peptide, and actually different compartments have different target peptide or signal peptide. And normally for chloroplast and mitochondria, the transit peptide exists in the N-terminal of the protein, but for nucleus, localized signal exists in within the signal of the protein. So by using some different bioinformatic tools, we can predict the subcellular localization. Actually, the bioinformatic tool that can be used for prediction of subcellular localizations, they have different strategy. Some tools depend on sequence features, so these tools analyze the protein sequence and extract features that are indicative of subcellular localization. These features can include amino acid composition, sequence motifs, sequence similarity to known localization patterns, and the presence of targeting signals or signal peptides. Some tools depend on homology. So homology-based methods compare the query protein sequence to a dataset to a data set of experimentally characterized proteins with known subcellular localization. By identifying homologous proteins with known localization, these tools interfere the localization of query protein based on the sequence similarity. Some tools depend on the machine learning. Some tools employ machine learning algorithms to predict the subcellular localization. They are trained on a dataset of proteins with known localization and learn patterns and relationship between sequence features and localization. The trained model can then be used to predict the localization of new proteins. Uh, Also, some bioinformatic tools combine these strategies together. So many tools combine multiple sequence-based features such as amino acid composition, physiochemical properties, and sequence motifs to improve the accuracy of predictions. So these tools often use sophisticated algorithms such as support vector machines, random forests, and deep neural networks to integrate and analyze these features. So I suggest you for prediction of subcellular localization to use different tools to obtain a good result. One of the important information can be written in the table is the molecular weight of a protein. The molecular weight of a protein is the atomic weight of all atoms in its chemical formula of that protein. Normally is measured by Dalton or kilodalton. So there is also some bioinformatic tool that can predict the molecular weight of any protein. One information can be written in that table is isoelectric point of a protein. So what is isoelectric point? Is a pH at which it carries no net electrical charge. It's determined by the presence of ionizable amino acid residues such as acidic and basic residues. Also, there is some bioinformatic tools that can predict the pI. Friend, I will show you now the table that exists in the genome-wide gene family identification shown in here in this paper. The first result is a table that exists this information. As I mentioned in the lecture, also there is another paper. You can check it. You can see that also it has a table that contains some information. Also another paper you can see. First, first result is a table that include information about the gene ID, amino acid, the molecular weight, isoelectric point, subcellular localization, and also the chromosome that where the gene exists and what is the group and conserved domain. So as last time we retrieved some protein sequences, DNA sequences, and cDNA sequences. Now we will use different bioinformatic tools to firstly to check really the sequence we obtained belong to the same gene family. So I will select one protein sequence and I will run uh, plus using smart tool. Smart tool actually is very important tool, can be used to predict the conserved domain in any protein. So how we can use it, just we search about smart tool. This uh, is a smart tool is a different option. No need to select any domain or so on. Just from here blast your protein sequence in FASTA format and click all these options. This is uh, include the database that can be used for searching and in your conserved domain. So run sequence smart. So we need to wait some time to show our result. So your job now is first position, and this website will be updated after 10 seconds. So here as it's showing that this protein has glycohydro 100 conserved domain, and I know this gene family should include this conserved domain. So by this way we can confirm the whether all our protein sequence was selected correctly. So after checking the protein sequences that obtained in last lecture, to have the same conserved domain, as I know based on our reading our and the references. So I will check the whether the cDNA sequences we obtained has a operating frame. How we can do it? We can copy the cDNA sequence uh, to operating finder in the NCPI. So plus the sequence in this box, and then select the minimal ORF length. There is several options. I select the longer one, and then select genetic code to be also there is several options. I select the standard one, and then select open ORF start codon to use ATG, and then submit. Wait some time. You can see here the sequence to predict one operating frame, and the operating frame is started from first nucleotide to last nucleotide. Also you can check the operating frame to have the same conserved domain. You can make a smart blast. Will open new window. So we need to wait some minutes. This okay. So result is now is available here, showing the conserved domain that exists in this ORF. So by this way you can confirm that the sequence you obtained. So after checking our sequences as have the same conserved domain, and also they encode ORF. So next we will obtain some biochemical properties like molecular weight and isoelectric point. How we can do it? We can use the protein sequence here and go to the uh, Swiss Institute of Bioinformatics. Actually, this website has a lot of tools can be used to obtain a lot of information. I selected one tool. It's a Pro Param. This tool is can paste here your protein sequence and this time just enter one protein sequence in signal letter code, so no need to use vasta format. So we remove this one. As I told you before, in every tool has different format to be used, and all these tools you can see the link in our video description, and then we compute the parameters and we wait. Now the result is showing here. You can see here some information. So the number of amino acid of this protein is 643. The molecular weight, as I told you before, is measured by Dalton, is almost 72 kilodalton, and the theoretical isoelectric point is 6.7. Also, you can get here some the composition of your amino acid, and also so by this way you can get some information about your protein property, and we will use molecular weight and isoelectronic point to make our table as I show you here. Finally, we will use protein sequences to predict the subcellular localization of our proteins. There is an important notice. We have to know that we cannot use DNA or cDNA sequences to predict the subcellular localization of our genes. So we have to use a protein sequence. Most of tools can use FASTA format. In this analysis, I will use 5 different tools to predict the subcellular localization of our proteins. Firstly, I will use target P. So I blast here my protein sequence, and I select the organisms. I am working in a plant, so I select plant, and then I select short output to be more faster. Also, I used localizer is a subcellular localization protection tools for plant and effector protein in the plant cells that can protect the proteins with chloroplast, mitochondria, and nucleus subcellular localization. So also I placed here my sequence. I select an option for plant sequence, and I run localizer search, and also I used methofates. This tool can predict the proteins with mitochondria signaling peptide. So also I use here I place my sequence and I select the organism plant and then submit the sequence. Also I used eukaryotic MPLOC two prediction tools to predict the subcellular localization of our proteins. This tool can predict the eukaryotic proteins with the different subcellular localization. So sometimes some protein can be predicted in different sites. So also I submit here my protein. Finally, I use WRF piece sort tool. So I select the organism I'm looking in plant, and then in this tool no need to use a FASTA format, just use the protein sequence. I remove here this one and run plus, and we will compare the result of 5 database. So target P can predict the protein sequence that localized in mitochondria, chloroplast, or cell wall. As is showing here, the most likelihood to be localized in chloroplast for this protein. Also, if I check another tool localizer, also is showing here, this protein has more potential to be localized in chloroplast. And if we check myotophates, we can see here this protein doesn't have mitochondria base sequence, so it doesn't localize in mitochondria. If we check here by using UKMBLOC, we can see this protein was predicted to localize in cell membrane, chloroplast, and cytoplasm. Also, if we use ORF tool, we can see this protein predicted to be localized in chloroplast, nucleus, cytoplasm, and plastid. So in overall, I can predict this protein to localize in chloroplast. However, we cannot make sure we by these tools, and we have to do some experiment. I will show you in next video how we do this experiment to confirm the subcellular localization of our protein. So I will try by another protein, this protein. So the same options we can use. We can compare the subcellular localizations by using different tools. So as I showing here that I'm using 5 different tools to predict the subcellular localization of our protein. So now we can check the result. So this protein predicted to have mitochondria transfer and also chloroplast transfer, and also by this tool also is predicted to have chloroplast transfer. By using this tool, you can see here this protein was predicted to have possessing mitochondrial sequence, so it's localized in mitochondria. By using this tool, this protein was predicted to be localized in cell membrane and nucleus. By using this tool, this protein was predicted to localize in chloroplast and nucleus and plastids. Overall, by using these 5 tools, I can predict that this protein to be localized in chloroplast and mitochondria. And so on, you can use all the protein sequences here to predict the subcellular localization, and we can make a table like this one. So I hope this video to be useful for you and bring some good information to you. And please like, and please don't forget to make share and like my video, and write in the comment if you need more information, and subscribe. And thank you so much for the watching.